The overall goal of this procedure is to measure the activity of Kv7 potassium channels in freshly dissociated mesenteric artery smooth muscle cells and relate that to the measurements of arterial diameter recorded under physiological conditions. This is accomplished by first surgically removing the mesenteric vascular arcade from a rat. The second step is to dissect and clean the segments of the mesenteric arteries. Next, some segments are used to isolate individual smooth muscle cells for patch clamp electrophysiological measurements of Kv7 potassium currents. The final step is the cannulation of intact arterial segments for pressure myographic measurements of arterial constriction or dilation in response to the pharmacological modulators of Kv7 potassium channels. Ultimately, the combination of patch clamp electrophysiology and pressure myography is used to show how an increase or a decrease in the activity of Kv7 potassium channels affects mesenteric arterial constriction.
The main advantage of this technique over existing methods, like the use of cloned channels expressed in cell lines, is that the currents are recorded from non-cultured cells that natively express the channels, and the Kv currents can be isolated from other classes of potassium channels that may be activated in the same cells. Pressure myography allows the functional responses to be recorded from the same tissue from which the isolated cells are derived, and the method is both more sensitive and more physiological than other preparations like arterial rings or arterial strips. Dr. Bharath, a postdoctoral fellow from my laboratory, will be demonstrating the arterial cannulation and pressure myography techniques. And Dr. Lioubov, a research assistant professor in my laboratory, will be demonstrating